like in a detailed view like mitosis. So we're gonna speak about the cell cycle, mitosis, cytokinesis, regulation of the cell cycle. So the cell cycle overview. The cell cycle what? Hey guys, give me a second. Why it's muted? Can you hear me, guys? You can hear me. Why say it's muted? Okay, there is a problem. It's showing that I'm muted. I don't know if you can hear me. I cannot understand. Okay, let me just make sure the chat. Yes, we can hear you. We can, yeah. Okay, perfect. I don't know why it says that I'm muted. If there is anything you cannot hear, just stay on chat. Okay, let's continue. So the cell cycle is a carefully controlled process that makes sure cells grow and divide correctly. Correctly and equally. It has two main stages, interphase and the mitotic phase. Now, if we're speaking about meiosis, it's interphase and the meiotic phase. So it depends on which cell cycle. Interphase is the longest stage where the cell gets ready to divide. It gets ready in a way in duplicating the DNA. It gets ready in a way to allow the cell to condense, the chromosomes to condense, to start to divide in a very good way and to align in a good way. And it's the main use of interphase is the S phase, which is like the second phase and which allows the duplication of the DNA. So, okay. So it has and mitotic phase includes nuclear division where the cell's nucleus splits and cytokinesis where the cell actually splits into two. And guys, you should understand that the sequence of the sequence of our mitosis starts with interphase, then mitosis itself or mitotic phase, and then it goes to the cytokinesis. The cytokinesis, usually questions are asked in the IMAT in a way that when does the cytokinesis happen during the mitosis? So the main question asked is before telophase, which is the last phase in mitosis, or after telophase. So of course it's after telophase, and the cytokinesis only happens in mitosis, not in meiosis. So cytokinesis. Mitosis and after telophase. They might try to trick you in a way to tell you that, okay, when does the cell divides, in telophase or in the cytokinesis phase? So of course here you have to say that it's in cytokinesis phase, the cell splits into two. Not in telophase. Telophase, the cell starts to split in this way, look. Cytokinesis, now we have two already cells. Okay, so you should like just make sure that you understand the cytokinesis thing. So, okay, cell cycle overview, we have G1, G2, G1S, G2. And then we have the mitosis and the cytokinesis. If you make a look, you would see that the longest phase is the interphase. And this interphase does not just start before mitosis. It can, it's not, it can, it is the main phase also in the meiotic phase. So interphase happens in both the mitosis and the meiosis. So in G1 we have the cell growth. Here the cell starts to grow, and then S, the DNA synthesis. So you should memorize in the S we have DNA synthesis and DNA replication or duplication. Duplication. So the Q material we have becomes 2Q in the S phase of interphase. And then interphase we have the second one is G2, which is also the cell growth. And then the mitosis and the cytokinesis happens, and of course the process continues and continues to happen and continues to happen because we need so much duplication of cells in our body and renewing of cells in our body us for us to be able to grow gradually. And one of the main things that they might ask you about mitosis is, so does it form four cells or two cells? It forms at the end only two cells, and these two cells are identical to their mother cell. And these two cells also are daughter cells. So these two cells are daughter cells, means are identical to each others and identical to their mom. So, or to the mom, to the dad, yani. So it can be like, here we have the mom. It made this two cells. Let's say the mom starts with an n number of chromosomes. They end up with an n number of chromosomes and an n number of chromosomes. So mitosis ends up with two identical cells. Okay, so both of them are identical to each others and identical to their mother cell. Make sure you memorize that there are only two cells. So like how it happens, how do we have so much renewing of our cells? The first cell makes two cells, or let's say the zygote. After a fertilized egg of a mom and a dad, of the somatic cells, I'm not speaking about the gametes, I'm speaking about the somatic cells. Somatic cells are all of the cells of our body, taking out all of the germ cells, which are the gametes, which are the sex cells. So somatic cells and the germ cells, these can be also named body cells, so body or somatic, and these can be named sex cells or also gametes. The somatic cells usually undergo mitosis, and the germ cells usually undergo meiosis. Okay, so let's speak about the zygote, about the baby when he grows and he proliferate in the endometrium of the mother cell or in the uterus. The somatic cells of this zygote undergo mitosis. So what happens? The first cell will make two cells. Then this cell will make also two cells. And then this cell will make two cells. But this shape does not mean this is meiosis. No, this is still mitosis. But look, here we have one. Here we have another mitosis. And then this makes two, and this makes two, and this makes two. So you have first to make sure what kind of cells you're working on to know each division, what is going on in the division. Is it a mitosis or a meiosis division? G1 phase or gap 1 phase is the first part of interphase. In this phase, the cell grows and performs its usual functions. It also makes important preparations for copying its DNA in the next phase. G1 includes key checkpoints that check the cell's DNA for any problems and assess external signals before moving on to the S phase. So you can say it's a kind of growth phase that makes sure everything is prepared for the DNA to be duplicated and copied in the S phase. So it's, we can give it a name, the preparation phase. Okay. The S phase or synthesis phase or the duplication phase is when DNA replication happens. During this phase, the cell copies all of its DNA, so each daughter cell will get a complete set of genetic material. So here, this S phase is the main phase of the whole interphase, and DNA replication or synthesis happens in the S phase. So what happens if we have one chromatid and let's say we have two chromosomes and we have one chromatid of each chromosome, it will be replicated, so each one will be a full chromosome having a condensed shape and having a two chromatids. Okay, so here we have the G1, then we have the S. If you see that we have the only thing that changes between the G1 and the S phase is the chromosome. They are being replicated. Okay, what kind of method our DNA? I want to ask something for it. Continue. Let me go to chat. Okay. Okay, I want to just make sure that everyone, that if anyone have a overview about this. So what is the method that our DNA use to be able to undergo the replication? What is the method? What's the name of the method? Does anyone know? Can you repeat? Okay, so what is the method that the DNA use? What is we okay, we have three methods, okay? So which one of the three methods that we have? I don't know because since most of you finished already the course, so I want to make sure if someone went through this. Okay, my voice is lagging. Okay, is my voice lagging for everyone? Anyone can hear me or you cannot hear me? For me, it's fine. Okay, Humera, try to change your place. Try to change your place for the Wi-Fi. You can hear me. Okay. So guys, which method does our DNA use to be able to replicate, to be able to do to undergo the replication, to be able to undergo the duplication of DNA? What is the name of the method? So I think no one knows. I'm gonna go and explain it. Excellent job. So semi-conservative method. Bravo, Rafi. Okay. So you should also memorize for the IMAT that this DNA replication or this DNA, this cell cycle, in which the DNA duplicate, the method of it, it's called the semi-conservative method. So we have two other methods, but we're not asked about them. You should not even know about them. You should know that our DNA, the method that we use is the semi-conservative. This memorize it, okay? So let's say we start with this. I have to use different colors. Okay, let me try to do this. Yeah, let me try to do this. So let's say we have one chromosome, okay? And this chromosome is already undergo the synthesis phase and it's already duplicated and this thing. So what do we have? We start with the two same chromatids. This is the same chromosomes. These chromatids used to be looking like this. Okay. So then the semi-conservative methods or the model of the DNA replication shows that first, after we have this, what happens here? It will be divided. When the division happens, we're gonna have one of them which looks like the mother or the same exact copy of this DNA of this chromosome, and the other will be no. Okay, let me try to draw in a better way because here it's so small. I'll go to the whiteboard. We're gonna speak about the semi-conservative and then okay, 1 second, let me stop sharing. Perfect. So the semi-conservative method, as we said, that we start with the two chromatids, and then they're gonna divide. Each one of them, it will divide. The first one is gonna divide in this way. Okay, here do we have perfect. And the second one gonna divide in this way. No. Okay, it's not working. Okay. Okay. So what happens in this? We're gonna, in the semi-conservative method or the semi-conservative model of the DNA replication, what happens here? We will have a new chromatid or a new new trait duplicated and a new allele expressed from the parents. However, this does not mean that we blocked or we stopped the old one of the parents. No, the old one will be there from the mom, from the dad, and it will be there. We will not like stopping. We're not stopping the expression of the parents' alleles or of the parents' material. However, we're gonna create a new material, and it will be in the DNA replication model. So the thing that we should understand is our chromosome will be divided in a way that we're gonna conserve half of our parents' material, and we're gonna create new material during this replication. So it's semi-conservative because it will halfly conserve parents' chromosomes. However, it will create other chromosomes or other chromatids. So we have other. Let me try to do this. So we have another kinds in which also our DNA is replicated, but before the new scientists knew that the replication happens in a semi-conservative method, they thought that it happened in two different methods other than this semi-conservative. However, the semi-conservative was the last method that the new scientists reached to know that, okay, how is the DNA replicated? But before, they used to think that it used to be conservative or dispersive. And they used to look like this. They used to be conserved, both of them, in the first one. However, in the second one, it used to be a very new alleles, but in the second one only, not in the first one. And the dispersive one used to be like halfly, halfly. So half a new trait, half old one. Half a new trait, half old trait. So they thought at the beginning that they used to be half by half, okay, or they used to be conserved in one, but in the other it's all of it new. But they reached the point that no, our DNA is replicated in a semi-conservative way. We're gonna conserve in each new cell, we're gonna conserve in each new chromosome half of our parents' chromosomes, and we're gonna create a new alleles and express them also. So they're gonna be half by half. So it's semi-conservative. The conservative and the dispersive, you're not asked about them for the IMAT, and they are not even in our body. However, the semi-conservative is the real one, which happens in our body in a replication of the DNA, and it will be asked in the IMAT. So the only model that you should know is the semi-conservative model of the replication of the DNA. Okay. No, Sayed, you're not yet. If you can watch the recording later. And we were just speaking about the interface. I think you know it. So okay, the screen. Okay. Okay. DNA replication is a very precise and carefully controlled process involving several enzymes and proteins. By the end of the S phase, each chromosome has been copied, creating two identical sister chromatids that are connected by a centromere. So here we have chromatid, the centromere, and the place or the loci or the locus in which these two exist together, it's called the kinetochore, in which these two make an intersection. So, S phase, we're gonna have a new synthesized strand after having the normal strand. And we're gonna, of course, have a leading strand and a lagging strand. The leading strand is this one in blue, and the lagging strand is the one in yellow. And then we're gonna it will use the DNA helicase and the DNA primase. Both of them together are called the chromosomes. And the topoisomerase to be able to undergo this duplication of the DNA. So by these filaments going inside the sliding clamp, we're gonna end up having the new duplicated DNA. Now, we should also know that this DNA polymerase will do something. Okay, so this DNA polymerase will do something on the leading strand and will do something on the lagging strand. So the DNA polymerase, let me draw it in a better way. Okay, 1 second. Because I think it's the kind of complex complex shape on this side. So I'm going to try to make it look better. Okay. So let's draw it here. So here we're gonna have, after the sliding one, we're gonna have two strands. We're gonna have the leading strand and we're gonna have the lagging strand of the DNA to be able to be replicated in the S phase. So here the leading and here the lagging. Which kind of enzyme working here is the DNA polymerase. What will it do? DNA polymerase main function is to create the Okazaki fragments. On each primer, it will create an Okazaki fragment. So what happens in this? So on the leading strand, we won't have so much primers and we won't have so much work for the DNA polymerase. It won't be creating this much Okazaki fragments. However, on the lagging strand, it will create so much Okazaki fragments to for the DNA replication to happen. So here we're gonna have the DNA, and then it will start to be replicated in a way, and the Okazaki fragments will be created on the lagging strand. So if you see on the lagging strand, we have so much Okazaki fragments, and they are made, of course, by the DNA polymerase with the help of the DNA helicase and the primase enzymes. The helicase, it usually helps in separating these two strands from each others. If you see, it comes in a way which having here like a very sharp top, and tries to separate these strands together. If you see, it separated this one from this one. So the work of the helicase to separate two strands. Okay. And the work of the polymerase is to create the Okazaki fragments. So, yeah, and I want to speak about something else. The topoisomerase, yeah. So the topoisomerase is the exact opposite of the helicase, and it tries to bring them together. So we have the helicase cuts them, the topoisomerase brings them together, and that's all you should know about it in the replication of DNA. Now we're gonna go into the mitotic phase. Now let me just go make sure that everyone is okay, because I want to also explain something on the board. So everything is clear? So before we go to mitosis, I want to explain something on the board. It's like something might be asked. And we're gonna speak about all the proteins that interfere in the cell cycle. So guys, protein. So one, we're gonna have cyclins, and number two, we're gonna have the vitamins. Cyclins and vitamins are one of the most, like, they'll be asking what is the role of the cyclin and the vitamins in the cell cycle. You should say that the cyclin is one of the main proteins that interfere in the cell cycle. They help in the cell cycle. So cyclin, cell cycle, from their names. So we're gonna speak about them because they are asked so much, and especially in the IMAT, not just in the IMAT, in the Italian exam, medical exam. So they might be asked in the IMAT. Because this year they in the Italian medical exam they asked so much about cyclins. So cyclins and certain vitamins play important roles in regulating the cell cycle. What are cyclins? Cyclins are a family of proteins that are crucial for the progression of the cell cycle, and they bind to and activate cyclin-dependent kinases, or the CDKs, let's say. They bind to them. And these CDKs, what are they? They are enzymes that drive different phases of the cell cycle. So because of these cyclins and these vitamins, they are kind of enzymes that allow the cell cycle to continue. These cyclins and vitamins, when do they interfere? In which part of the cycle they interfere? They interfere between the interphase, and mitosis. So they happen here. When the G2 phase finishes, or let's say G2. When the G2 phase finishes, the moment that they are entering the mitosis to start the mitosis, here the cyclins interfere and the vitamins interfere. So between the G2 and the mitosis, these two help. So as we said, they bind on the CDKs to start, or on the kinases enzymes to start the cell cycle. The cyclins you should know for the IMAT are cyclin D, cyclin E, cyclin A, and cyclin B. Cyclin D is involved in the G1 phase, helping cells prepare for DNA replication. So it helps in cyclin D is one of the non-important cyclins in our cell cycle, but we have to know that they just help in the G1 phase or the growth phase or the preparation phase for the DNA replication. So they just prepare for the DNA replication for S phase, let's say. Cyclin E helps cells transition from G1 to S phase where DNA replication occurs. So they just help in the transition from G1 to S. So we can say almost D and E are together, but of course D starts before the E. Cyclin A, it regulates the S phase and the transition from S phase to mitosis. So, or the M phase. And before mitosis, there's something called the mitotic phase or the M phase. So we can say instead of mitosis itself, we can say M. So A, it regulates the S phase. So it regulates the S phase and the transition from S to M, or to this phase. M phase means the phase which is before the mitosis, or we can call it the pre-mitosis. Cyclin B, and why did I write this here? Most importantly, because I want to speak about cyclin B. Plays a central role. So the most important cyclin in the cell cycle is this, and the most asked one is also cyclin B. So focus on cyclin B. It plays a central role in driving the M phase and bringing the M phase, where cell division takes place. So, of course, we start from the least important to the most important here. Yeah, from the least to the highest importance and the most asked. So one of the least important are D, which helps in the preparation of S phase, and then vitamin cyclin E starts working from G1 to S, and then vitamin cyclin A regulates the S, and then B brings or plays a very central role in driving the cell cycle. So these cyclins, you should know them. Starts with D, E, A, B. However, you should focus on the B cyclin, and you should know it's the main cyclin that helps in the cell cycle to start. Okay, now let's start to speak about the vitamins. So not just the cyclins that help us in making the cell cycle occur, however, also there are some vitamins that also help us in allowing the cell cycle to occur. So certain vitamins have been found to influence the cell cycle and cell division. Vitamin A, vitamin C, vitamin E, vitamin B9, and vitamin D. So the main vitamin that you should know from all of these vitamins for the IMAT is vitamin E. The main cyclin you should know for the IMAT is cyclin B. Okay? So let's start. So vitamin A, and so it's involved in regulating cell growth and differentiation. It can promote cell cycle arrest and apoptosis in some cancer cells. Repeat B, please. Cyclin B. Okay, so cyclin B, yeah, sad. It's a cyclin or it's a protein that is involved and that plays a very huge role in driving the M phase, where cell division takes place. When the mitosis starts to happen, but from G2 to M, between these two phases, cyclin B here comes and plays a role in allowing the M to happen, allowing the mitotic phase to happen. So cyclin B, they will ask you, what is the main cyclin in all the cyclins to allow the cell cycle to happen? You will say cyclin B. This is the only thing that might be asked about them. However, others, you should know them. No one knows what happens to their minds. They might ask, they might not ask. This year, no one knows. And the same of vitamins, you have to know them all, but like focus on vitamin E. They might ask about the others. No one knows what happens to them. So, is it clear, Saad? Okay, I think it's clear. Perfect. So the vitamins. Vitamin A, you're welcome. So vitamin A, it's involved in regulating the cell growth and differentiation. It can promote the cell cycle arrest and apoptosis. So with that, cyclin A, it helps in also driving the mitotic division and the replication and everything. However, there is something additional in A is about apoptosis. Okay, do anyone know what does apoptosis mean? Programmed cell death, exactly. So apoptosis is the programmed cell death. It's controlled cell death, exactly. So why does apoptosis happens? Why do people do it? Why do humans do it? It's a kind of technique that we do to be able to kill cells that are unneeded or affected cells that are unneeded for the body or for anything. So vitamin A plays a very good role in the apoptosis. So they might ask you about which vitamin helps in apoptosis. You can also say vitamin A, other than the replication. Vitamin C. Okay, so of course, vitamin A helps also in the replication, but not as vitamin E, so you don't have to really focus on this. Vitamin C acts as an antioxidant and can help support DNA repair mechanisms, potentially mitigating cell cycle arrest and genomic instability. So it just also helps in the cell cycle in a way that can support the DNA repair mechanisms or can support also the replication. So vitamin A, vitamin C, both of them are not the main ones, but they help in the replication. Vitamin E, which is the most important, another antioxidant. So these two are antioxidants that may help protect cells from oxidative stress, induce cell cycle disruptions. So let me just write the vitamin E role because you will be asked about it. So vitamin E, or other name is tocopherol. So it's antioxidant, vitamin, help in protecting cells from oxidative stress. Induce cell cycle disruption. So if we don't have vitamin that helps in the protection of the cell from any stress around it, from any stress in the surrounding, from any oxidative stress or chemical stress which is induced around it, what happens? This cell won't undergo this mitosis or this cell cycle. So one of the main vitamins that helps of the cell cycle to continue is vitamin E because it helps very, very main function in the protection of this cell to be able to continue its role. Because very high probability that this cell might die in any way from any kind of stress around it. So vitamin E is the helper here. Vitamin B9, another name is the folate. You don't have to know the names, I'm just writing them. So this vitamin B9 just helps in also the replication, and vitamin D also the same thing. And yeah, it also helps in the protection from oxidative stress induced cell cycle disruption. So I think vitamin D and vitamin E are the main vitamins you should know. Vitamin A, vitamin C, vitamin E, and no, vitamin A, vitamin C, and vitamin B9. You should just have them as additional information. Just focus on vitamin D and vitamin E, and focus on the oxidative stress induction. So these are the main things we should know for the IMAT about cyclins and the vitamins, nothing more. Vitamin D and vitamin B9. Vitamin B9, nothing. It just helps in the replication. It's just like there for the replication to happen, nothing else. Like does not have a specific role for it to happen. Vitamin D is the same exact function of vitamin E. So D and E are the same. And vitamin B9 is the same as A and C. A is only for apoptosis, is usually used in apoptosis, and yeah, that's all. And also vitamin D can be used in some cancer cells. So you can add it to this also. So D, E, and C are antioxidants. Yeah. They help in protection against the oxidation stress. Okay, perfect. So now we're going to continue the slide. Okay. So now we spoke about the cyclins, we spoke about the vitamins, and we spoke about interphase, and now we're going to go for the mitosis itself. So the mitosis, as I said, here the interphase from here to here, this is the interphase. In G1, we start growing the cell. In S phase, the chromosomes are already duplicated, and in G2, it's all grown cell and ready to start the cell cycle. In prophase, so we have here prophase, prometaphase, metaphase, anaphase, telophase, and then cytokinesis. So here happens the cytokinesis. Perfect. So what happens in prophase? Prophase, as we said, that the chromosomes start to be condensed. If you see here, they were looking like this, and then they started looking like this, super condensed, and they are not chromatin shape, they are really chromosome shape. So, and then here the nuclear membrane starts to disappear, the spindle fiber starts to form, all of these things happen. And if we remember, we spoke about the spindle fiber, and it's a kind of microtubules in our body that helps in the structure, number one. And number two, it has a function of the cell cycle. So this like spindle fiber. So here we have this, as we said, the chromosomes are condensed, spindle fiber starts to form, nuclear membrane start to disappear. Here we don't have no more nuclear membrane. The centrosomes created already a spindle fiber. These two are called centrosomes. Centrosomes. And then the spindle fiber is already formed, and the chromosomes are aligning together. The homologous chromosomes are aligning together. Prometaphase is just this, that they just align on the spindle fiber. However, in metaphase, they divide. One will go up and one will go down. So here we have an orientation of the chromosome. In anaphase, we have sister chromatids align on the equatorial plate, so or segregate on the equatorial plate. So here we will have a division of each chromosome into two chromatids, chromatid and chromatid. One chromatid will be up, one chromatid will be down, and this is anaphase. And at the telophase, we're gonna have two cells, each cell having one chromatid from each one in this way. And if you see, we ended up with a semi-conservative method of um, we ended up with two cells having the same shape of the mother cell. Who is the mother cell? Here is in G1, this one. So this is the mother cell, and these are the two cells or the two daughter cells, and super identical to the mother cell from the shape, from this number of chromosomes, from everything. So in telophase, if you see, it is almost dividing, but it's not divided yet. And it only divides in the cytokinesis to be able to give the two cells, each one of them having the same number of chromosomes of the mother cell. Now, there's something that I want to speak about, the telophase and the cytokinesis. So let me see the whiteboard. Chat. Oxidative stress leads to necrosis. It leads to the explosion of the cell. And of course, I'm speaking about the plant cell. Because usually plant cells, when they are super filled in water, like they have very high pressure of water around them, the cell cycle does not really happen easily. So they really need the antioxidants. Okay, so here I want to speak about the eukaryotic cells. So as we spoke before about the eukaryotes and the prokaryotes. So the eukaryote and the prokaryote. The prokaryotes, I'll speak about them after finishing the eukaryotic cells and everything about the cell cycle. So we have to leave them. The eukaryotes, who are they? Are the plant cells or the animal cells and the plant cells mainly. So in the eukaryote, after the telophase, we have a cleavage of two daughter cells. This cleavage is the cytokinesis. Now, how this happens? Yeah. So how this happens? In the animal cells, we're gonna have two, let's say if you remember now in the slide, we had a structure looks like this. Gonna have here chromosomes, or like one chromatid, it's the same. So how the cytokinesis happens, it will start to cleave it to end up with a cell. So this cleavage of the animal cell is called the furrowing. Furrowing. It will end up with two cells. However, the cleavage of plant cells, it will be like this. Because you know the plant cell have a cell wall, so it will be different structure than the animal cell. So we're gonna have inside here and it will start to cut here the it will start to cut here the two cells. However, something will happen. It's called the cell plate formation. And the vesicles are gonna line up here, and we're gonna create a new cell wall. The shape will be like this. So a new cell wall. Why a new cell wall? Because if we divide them together, if we didn't create a new cell wall, if we didn't have the vesicles lined up here, what will happen? The two cells will end up like this. And the second one will be like this. So we won't have a cell wall here. It won't be a full cell or full grown cell. So that's why we're gonna undergo the cell plate formation, and the new cell wall will be created between them before these two are cleaved. Because if we cleave them and there was no cell wall here, what happens? As we said, we're gonna end up without a cell wall of each cell. So we have to create it before we cut it. And then here the vesicles will fuse together. So we're gonna have vesicles line up, and then the vesicles will fuse together. This is like the formation of a new cell wall, and this vesicle is working. It's called the cell plate formation. However, the division or the cleavage of the animal cell is called the fuse. So this is what you should know about the cytokinesis. And yeah, any questions? I don't think so. So I was speaking about the kinetochore, and I just want to explain something about it. So I said that we have two chromatids here, and we're gonna have here the centromere. So here we're gonna have also the kinetochore. This kinetochore, where is it? It's next to the centromere. Okay, there's no space. So what does it work? It splits the chromosome to two sister chromatids in the anaphase, in anaphase 2. Or in anaphase, let's say, since we are still in mitosis. So the kinetochore main role is to split the chromosomes. It exists here, and when the chromosomes need to be splitted and to be formed as chromatids in anaphase during the cycle, it will work as a splitter for these two chromatids. And by the way, one chromatid is called a chromosome. Even if it's just a chromatid, it's still called a chromosome. And I want to say something else. So what you should also know about the mitosis. So we're gonna start. So I'm gonna start with the mother cell. Let's say it has a 2n. Since the why 2n? Because in the S phase the n is duplicated. So in the S phase of interphase, it used to be n, it became 2n. And then it will undergo, it will end up with two identical cells. So when we undergo the metaphase, and then if you see here in the anaphase, it will look like this. And the equatorial plate will be like this, like this, like this, like this. So here you should remember that in the metaphase, homologous chromosomes align or separate. And here we're gonna have sister chromatids align. So also you should know this definition of metaphase and anaphase, because usually they ask sister chromatids for anaphase, homologous chromosomes for metaphase. Okay. Okay, is everything clear to continue the slides? Okay. So, mitotic phase. The mitotic phase includes two main processes. Mitosis, this is the division of the nucleus. Cytokinesis, this is the division of the cytoplasm. These processes make sure that genetic material and cytoplasmic components are evenly distributed to the daughter cell. So these are the mitotic phase. As we said, the prophase, chromatin condense into visible chromosomes, nuclear envelope breaks down, centrosomes move to opposite sides of the cell, microtubules or spindle fibers begin to form. Anaphase, sister chromatids separate at the centromere. They move to the opposite end of the cell, helped by the shortening of microtubules. Chromosomes break at centromeres, and sister chromatids move to the opposite ends of the cell. When they break, who will be helping here in the breaking is the ketopole. Centromeres are what holds the sister chromatids together. Telophase, the separated chromosomes reach the cell's walls. New nuclear envelopes start to form around each set of chromosomes. Of course, because we're gonna have two cells, so we're gonna have two nucleus surrounding the chromosome since it's a eukaryotic cell. The mitotic spindle breaks down and chromosomes begin to unwind and decondense. The overview, we're gonna have the interphase, the prophase, and then the pre-metaphase, the metaphase, the anaphase, the telophase, and the cytokinesis to end up with two daughter cells. The cytokinesis happens after mitosis and it involves the splitting of the cytoplasm. In animal cells, it happens the furrowing, and here you see cleavage furrow. Okay. A ring made of actin and myosin filaments forms around the center of the cell. The ring contracts, creating a cleavage furrow, then eventually divides the cell into two daughter cells. So when we reach the muscle system, like the contraction of our muscles in the body, we're gonna speak about the filaments, which are the actin and the myosin filaments, and we're gonna know how do they work together, how do they like co-work together in order to break something or to allow us to move or to contract the muscle. So here, how does the muscle contracts or how does the cleavage happens? So how does the cleavage happens? Here we're gonna have the actin and myosin filament working here, and then by contracting together, they're gonna cut the cell. About the actin and the myosin, we're gonna speak about them later. In plant cells, a structure called the cell plate forms between the two daughter cells or the two daughter nuclei. The cell plate grows outward, merging with the cell membrane. It eventually forms a new cell wall, dividing the parent cell into two daughter cells. So we spoke about this also. And we spoke, I just drew this for you. So we have the cell membrane, we have the furocleavage, and we have the ring of the actin myosin. And telophase, a contractile ring. Yeah, that's all. We spoke about them. So cytokinesis in animal cells. Here we have the actin ring, and here we have the cleavage furrow. Here, furrowing, and here we have the cell plate, and here we have the cell wall. Of course, why? Because it's a plant cell. Regulation of the cell cycle. The cell cycle is a carefully controlled process. It ensures proper cell growth and division. Checkpoints monitor the cell cycle at specific stages. Verify DNA integrity. Ensure completion of essential processes. Regulatory proteins control the cell cycle. Cyclins and cyclin-dependent kinases, CDKs, are key examples, and vitamins. coordinate transitions between cell cycle phases. Regulation of the cell cycle. So we're gonna start with the G0 phase, G1, G2, S, G1, S, G2, and the mitotic phase. Yeah. And we were speaking here about the between the M and G2 here, the cyclin B gonna work. Or not just here, we can say from here to here. Here the cyclin B works. And the vitamin E and vitamin D. Okay, so MTOC. So we're gonna have a mother centriole, and we're gonna have here the microtubules around the cell, and pericentriolar material, PCM, and distal appendages. Okay, this you won't be asked about in the IMAT. So it mainly just speak about the microtubules. And if you remember, when we used to speak about the microtubules, we spoke about the grams, the positive and the negative grams. But you don't have to know about all of this. So here we're gonna have the interphase. Both of them have the centrosome and the centriolar satellite, and of course the microtubules. If you remember when we spoke about the microtubules, we spoke about the positive grams. So if you see how does it look like from, if we zoomed in, it looks like having positive grams here around. These grams allow the attraction of the chromosomes. Why is it important? Proper regulation of the cell cycle is crucial for maintaining normal cell function. This regulation of the cell cycle can lead to uncontrolled cell growth and division. Uncontrolled cell growth and division can result in conditions such as cancer. Understanding and who works on cancer? It's vitamin E. Understanding the intricacies of the cell cycle is essential for developing targeted therapies. Targeted therapies and treatments can help address various diseases related to the cell cycle dysregulation. So here you're gonna have the summary, and that's all the slide. So does anyone have a question? Okay. Yeah, homologous chromosomes in mitosis. And they also exist in meiosis. So does anyone have any question? Okay, one moment. Okay, one moment, 1 second. Yeah, when I said vitamin D is for cancer cells, it helps when we spoke about vitamin D and we spoke about all other vitamins, we said they might be antioxidants and they help for the cell cycle to continue. So if this problems happened, cancer might happen. So when the vitamins work, cancer might. About the homologous chromosomes, you were asking me if it's mitosis or meiosis. It's just a meiosis. Yeah, exactly. Yeah, just a meiosis. Let me just change it. Did I write it somewhere? But you will find that in the metaphase, you're gonna find also the chromosomes separating, but you cannot name them homologous chromosomes. I will send something on the group. You can check it. You can read it and know the difference, okay? Okay, so there is a new girl. She enrolled in the class like lately. I don't know if she's inside. Just have to say hello. No, she's not watching. Okay, perfect. So now we're gonna speak about the meiosis. So I guess, yeah, I gave you a note. Let me just try to change it or correct it. So when we spoke about the metaphase and the anaphase, I told you like here we're gonna have a homologous chromosomes and here we have a sister chromatids. Write this for meiosis, not mitosis. This for meiosis. Just change the word into meiosis, not mitosis. Okay. So now we're gonna speak about the meiosis in a simple overview. Okay. So meiosis, as we said, it just happens in the germ cells or gametes. And this meiosis is the exact same. It will undergo the interphase, it will have the cyclins, everything is the same. The only difference is it is made up of two phases, not just one phase. Meiosis 1 and the meiosis 2. So it will start up by one cell, and then it will divide into two. And then the second one, it will divide into two, and it will end up with four cells. These four cells are haploid and are not diploid. Why? Because it's sex cells, and sex cells usually are haploid and not diploid. Means they just have one chromatid or one chromosome. And what else we should know? That the first, this is meiosis 1, this is meiosis 2. The first meiosis, this one, is called reductional. And the second meiosis, this one, is called equational. Okay? How reductional, how equational? Now I will explain. So reductional means it reduces the number of chromosomes. Okay? Equational means it reduces the quantity of DNA by 2, not the number of chromosomes. Let's say we started with the n chromosomes. And the reductional here we're gonna have n over 2, n over 2, and here they're gonna be identical. So n over 2, n over 2, n over 2, n over 2. However, the quantity of DNA, we're gonna start with a Q. This is the quantity, so n, number of chromosomes, and the Q is the quantity of DNA. Should know how to differentiate them. So here we're gonna have a Q, here we're gonna have a Q, and here we're gonna have a Q over 2, Q over 2, Q over 2, and the Q over 2. So n, first the meiosis is n over 2, second meiosis is the same, n over 2, it's equation. However, the Q, the quantity of DNA is gonna be the same in the first meiosis, but reduced in the second one. I'm gonna try to draw a figure to make it more clear for you. Let's say this figure or this graph. Let's put here the quantity of DNA and Let's see. So it will be like this, and then it will repeat the same thing. So here we will have, let's do this. Okay. So here we have G1, here we will have S, here we have G2. These are the interface. And then here we're gonna start with the metaphase. I think in this way is better. Here, this is meiosis 1 and this is meiosis 2. So meiosis 1, prophase and metaphase, anaphase, telophase, and then we'll go to the meiosis 2, prophase 2, metaphase 2, anaphase 2, and telophase 2. Here and here we're gonna end up. What happens? So first we're gonna have the number of chromosomes. Number of chromosomes that start with an N. I mean, we're here working on the quantity of DNA. So we will start by Q. It will be here a Q. And then it will be, so let's put the small figure here. Q, 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 Q over 2, Q over 2, Q over 2, Q over 2. So meiosis 1, it will be Q. So we will stay on Q. This meiosis 1, we're gonna stay on Q. And then the meiosis 2 here, it will drop to be Q over 2. Here. Q over 2. Okay. So the graph says that when we start by Q, we're gonna have Q in the meiosis 1 and Q over 2 meiosis 2. If let's say we have the number of chromosomes instead of Q, the graph will be like this. So we have, so N over 2. When is N over 2? In the meiosis 1. So here meiosis 1 will be N over 2. And meiosis 2 will be still also N over 2. So all of them, it won't be looking like this. This graph is only for the quantity. It will be looking in another way. It will be looking in, let's say, okay, here will be N over 2 for meiosis 1 and for meiosis 2. So they will be together 1 plus 2 here. So here, if we're gonna talk about the number of chromosomes, but for the IMAT and for any exam, you just ask about the graph of the quantity of DNA, which just says that in meiosis 1 it will stay the same, and in meiosis 2 it will be divided into 2. The number of chromosomes, they divide from the first one, but they stay the same the second one. Okay, so meiosis usually happens in sex cells. What are the phases of meiosis? Here is the quantity of DNA. Quantity of DNA. Okay. So meiosis have something which is more like additional to than mitosis. Is called the tetrads. So the tetrad, okay, let me try to draw it. Draw phase 1. Okay. So in prophase 1, we're gonna have something called the tetrad. And then we're gonna have in metaphase 1, um, also the spindle fiber and the chromosomes will be aligned on the spindle fiber. However, the tetrads will also exist in metaphase 1. However, when does the tetrads exist? When they start to appear in prophase 1. So you don't have to make a confusion between them. Tetrads start in prophase 1 and not in metaphase 1. However, they still exist in metaphase 1. This does not mean that they don't exist. They're gonna exist all over the cycle. And especially the first cycle. So first we're gonna have the interphase, and then we will have the prophase 1, nuclear envelope disappears, and the chromosomes the spindle fiber will start to form, and the tetrads will happen. After this, the crossing over will happen. So from the prophase to the metaphase, we will have the crossing over. How does crossing over happens? Let's say we have two chromosomes here. And the crossing over usually happens between two homologous chromosomes. So crossing over is between two homologous chromosomes. So what happens in the crossing over? Let's say this chromosome have an A, this chromosome have a small a, this chromosome have a B, and this small b. So this A, this one will go with this one. Why it's not writing? So this one gonna be with this one, and this one gonna be with this one. So they will be like this. A, B, and then B, A. This is called the crossing over, and it usually happens from the prophase 1 to the metaphase 1. And then after this, after we have the chromosomes already aligned in the metaphase 1, the homologous chromosomes, we're gonna have in the anaphase 1 the chromosomes going on the equatorial plate from place to place. So here we're gonna have this chromosome in this way, and this chromosome. Try to draw it better. Here one. And after this, the telophase, we'll end up with two cells. Of course, they are not fused yet. And we will have the tetrads and the chromosomes together inside. And then we're gonna have also the cytokinesis. Now, after this, we're gonna have the prophase 2. We're gonna start, how does the prophase 2 happen? We will take each one of these and it will happen. How is this? We're gonna divide these two by the cytokinesis and by the actin filament and meiosis and whatever. And each one of them, it will undergo the division again. So let's speak about this one here. It will undergo a very, very normal cycle. The nuclear envelope starts to disappear because we already have a new one here. This is the nuclear envelope. And then here we're gonna have them. And then they will align on the spindle fiber. In anaphase, they will sister chromatids. Sister chromatids align oppositely on the plate. And then telophase 2 and the cytokinesis. This exact one will start also to divide and to have the cleavage, and we will have two chromatids, and this will be a haploid cell. So here we will start with the diploid cell, and we will end up with a haploid cell. Diploid means this chromosome. Haploid means this chromosome with one chromatid. The same thing will happen with the second one, which is this, the same exact thing, and we're gonna end with also 2 haploid cells, ending up with 4 haploid cells. So what you should know about it is that in meiosis, in metaphase 1, homologous chromosomes align on the spindle fiber. Anaphase 2, sister chromatids align on the equatorial plate. So here you should take it. You should take the note that metaphase 1, homologous chromosome. Anaphase 2, sister chromatids. Okay. So the shape of meiosis is we start with N over 2, 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 N over 2. This R4. This is called haploid cell. And what does haploid cell mean? Means it's either a sperm or an ovum. And this is called a diploid. No, not here. By the beginning, this meiosis 1, this meiosis 2. By the beginning of meiosis 1, here, I mean by the end of meiosis 1 means by the beginning of meiosis 2, so beginning of meiosis 2, we're gonna start with the diploid cell. And of course, this concept of meiosis is gonna lead us to the concept of spermatogenesis, and oogenesis. We're gonna speak about them later, but the main concept is meiosis. So why we're gonna speak about them? Because sperm and ovum are both of them are gametes, and of course they are the product of meiosis cycle. Beginning of meiosis 2 is from haploid. No, it starts from diploid. You're gonna end up, so in meiosis 1, let me write for you something. So in meiosis 1, you're gonna end up with a shape like this. Okay? It will have a chromosome with 2 chromatids, a chromosome with 2 chromatids. And then this, you will start the meiosis 2. So this will be cleaved. And here you're gonna have one with X, X. And here you're gonna have one with X, X. So we started with a diploid shape of chromosome. How are you sure about it? Meiosis 1, you start with a diploid. And also, meiosis, you said meiosis 1 or meiosis 2? Let me make sure what you said. Meiosis 2. Yeah, it's meiosis 2. Okay. Okay, guys, are you taking, are you screenshotting them? Okay, someone's spamming. Okay, so the resource I'm taking from is wrong. Okay, so meiosis 1 is diploid, and meiosis 2 starts with the haploid cell, 2 haploid cells. The reason I wrote diploid, and I was also so I was also like doubting, Is because that the resource, the book I'm getting from is wrong. Like it's not wrong, but I don't know why it wrote deployed. And I was also doubting this. So I will send you something on the group. If you see, it's written deployed. So you can use this exact same form, like the exact same paper I sent, but just take off the deployed and write haploid. Because we're gonna start with 2 haploid cells, not 2 diploid cells. So I'm sorry guys, it's just the resource. And thank you so much, Sayed. So guys, don't take it diploid, change diploid to haploid. So we start with the diploid from the beginning of meiosis 1, and then in meiosis 2, we're gonna start with 2 haploid cells, ending up with 4 haploid cells. Okay. So anyone have any question? No. Perfect. So now we still have 30 minutes, and we're gonna take a 5 minutes break, and then after this we're gonna continue, of course, the notes. You remember I used to give you notes at the end of each session, so we're gonna use this 30 minutes for notes.